Myself, Dr. Samakshi Verma, Assistant Professor, Department of Biotechnology, Rama University. Today I am going to deliver a lecture on cell division. Cell division takes place in basically both plant and animal cells depending upon the cells in which cell division is going on. So, depending upon the cells, so we are talking about, when we are talking about a cells, then there are two types of cells, somatic and reproductive. Somatic cells where the cell division occurs mainly for the cell growth, responsible for cell growth. But in case of reproductive cells, the cell division occurs to form the germ cells or gametes for reproduction process. Then after cell division, they will undergo fusion to form diploid structure. So, basically cell division undergo by cell cycle completes within two phases, that is M phase and interphase. M phase is mitotic phase, interphase the rest phase where the synthesis of DNA or the gap between one mitotic division to the another mitotic division, that phase is known as the interphase. So, the interphase is divided into four phases, G1, S, G0 and G2. G1 is gap 1 phase, S is synthetic phase, G0 is resting phase and G2 is gap 2 phase. This is a long interphase between a one mitotic to second mitotic division. But there is also a resting stage between meiosis division. Meiosis is basically of two types, meiosis 1 and meiosis 2. The rest stage between meiosis 1 to meiosis 2 is interkinesis, which is very short lived, where DNA replication takes place. So, cell division is basically the division of cell and the division of cell by nuclear and cytoplasmic division. When the cell undergoes nuclear division, then it is known as karyokinesis. When the cell undergoes cytoplasmic division, then it is known as cytokinesis. So, cell division is divided into two types, that is mitosis and meiosis division. Mitosis division, that is known as equational division because the number of chromosome between parent and daughter cell are same and the formation of identical daughter cells are seen in case of mitotic division. But in case of meiosis division, this is reductional division because in meiosis 1 number of chromosome becomes half with their parent cells. So, it is reductional division. First, mitotic division is divided into four phases, that is prophase, metaphase, anaphase and telophase. In prophase, we have already studied everywhere what is cell division. So, I am not going to tell you about the very detail. Here we are talking only about overview of these phases. In prophase, we have seen that there is a condensation of DNA material or chromosome is seen basically, and the nuclear membrane start disintegrated. In metaphase, the condensed chromosomes are arranged at metaphasal plate or at equator of the cell through spindle fibers attached to the centrosome. And then in anaphase, this division of the chromosome which are attached arranged at metaphasal plate in metaphase are now stretched and divided into two sister chromatids. The chromosome is divided into two sister chromatids and each sister chromatid pull towards opposite poles through spindle fibers attached through the centrosome, which are forming U to V shaped structure like this, U or V shaped structure in anaphase. After that, in telophase, the regeneration of nuclear membrane occurs and these two oppositely arranged opposite poles पर जो attached chromosomes हैं will move into different cell by forming a nuclear membrane in telophase stage and divided into two cells. So, this process of from prophase to telophase is karyokinesis process in which nuclear division takes place. So, the nucleus is divided into two types. After that, cytoplasmic division will take place, that is cytokinesis, and one cell is divided into two cells having same number of chromosomes in their daughter cells. These two daughter cells are identical to their parent cells having same number of chromosomes. Okay? And the mitotic division is generally known for genetic continuity, means the genetic changes or the genetic variations that is seen in parent will also be seen in their daughter cells. So, the genetic continuation or continuity will be seen in mitotic division. So, next is meiosis. Meiosis is divided into meiosis 1 and meiosis 2. Meiosis 1 is reductional division where the number of chromosomes in daughter cells is half as compared to their parent cells. And then the next is meiosis 2, which is generally the mitotic division as same as this. Okay. So, now the meiosis 1 is divided into 4 phases: prophase 1, prophase 2, prophase 1, metaphase 1, anaphase 1 and telophase 1. So, in case of meiosis division, the prophase 1 stage is completely larger, which has 5 sub-stages also, that are leptotene, zygotene, pachytene, diplotin and dikinesis. These 5 stages are only seen in meiosis 1, prophase 1 stage. So, the formation of bivalent complex or synaptonemal complex is seen in zygotene stage due to the enzyme recombinase, which is produced in pachytene stage, which helps in formation of bivalent complex or synaptonemal complex. In the leptotene stage, there is a disintegration of nuclear membrane and condensation of DNA material starts, and then in zygotene stage pairing of homologous chromosomes starts, and after that non-sister chromatids will recombinate to form synaptonemal complex. They are attached through the point, two sister chromatids are attached at a point, that point is known as chiasmata. Then in diplotin stage, the crossing over process takes place where the genes will transfer from one sister chromatid to another non-sister chromatid due to the formation of synaptonemal complex. And in dikinesis stage, there will be fully disintegration of nuclear membrane will be seen. Then in metaphase 1, in meiosis 1, metaphase 1 phase, we will see these exchange gene will arrange at metaphasal plate as seen in mitotic division. After that, anaphase 1, here we will see the exchange chromosome will separate into two sister chromatids having different genes because of prophase 1 stage will arrange at different or opposite poles in U or V shaped structure. After that, the last phase will be telophase. Telophase 1, in which the nuclear membrane start appearing and the chromosome start to convert into chromosome will divide into two different cells. So, the nuclear division is completed here, but unlike mitosis, here number of chromosome will be half in meiosis 1 division. After that, it will go meiosis 2 division, in which the one cell is divided into two cell having same number of chromosome. So, in case of meiosis, the number of chromosome is haploid as compared to their parent cell. So, we can say the mitotic division is equational division where the number of chromosome is same, but the meiosis division is reductional division where the number of chromosome is half or haploid number of sets, and the mitotic division will form 2 daughter cells, but meiotic cells will form 4 daughter cells. These 2 daughter cells are identical, but these 4 daughter cells are non-identical to their parent cell. This is the difference between mitosis and meiosis. So, this is the whole process of cell division that I have told you today about. So, here now I am ending my lecture with this. Thank you very much.